From Microbe TV, this is a special episode of Immune, an immune booster, recorded on March 14th, 2025. I'm Sydney Leifer, and you're listening to the podcast about the body's defenders against disease. I'm here in Ithaca, New York at Cornell University in my office. Today I'm flying solo, and I'm here to do a special interview, and I'm calling these office interviews. And really the goal is to learn a little bit, get a glimpse into the scientist behind the science. If you enjoy this and the other shows of Microbe TV, please consider supporting us. We are a 501c3 and all of your donations are tax deductible. You can donate at microbe.tv/contribute. So it's my great pleasure today to have a special guest, my first guest for an office interview, Dr. Shane Crotty from the La Jolla Institute for Immunology. Welcome.
Thanks for having me, Cindy.
So I've been following your stuff for a while. I was really excited to have you on this show, especially the first episode. You're here today to give a talk at Cornell and probably going to talk about your recent paper, at least some of it, in Nature on the upper airway, mucosal immunology. We did talk about it briefly on a recent Immune 85. Good. So and we did it as a snippet. So there's lots more to learn about that, I'm sure. And I know you've been on TWIV a couple of times, so it's pretty fun. And it's great to have you on Immune now.
Yeah. Where do you want to start?
So I thought that it would be interesting to ask you first, like, how did you get into science? And more specifically, when did you know that you wanted to be a researcher? Was it always, or was there a specific event or a series of events or experiences that led you to realize that this is your path?
Yeah. So when I was in high school, in my opinion, that there wasn't really any way to know you wanted to be a biologist or a molecular biologist because there was no science like that you could get exposed to, right? It was just textbooks.
Right.
Yeah. And so unlike math or physics or chemistry, there would be something you could get your hands on and actually, learn how to be an experimentalist, and that wasn't the case for biology. I was always very interested in science. I had the good fortune of there was an NSF Young Scholars Program at the time in Los Angeles. I lived in the desert, way outside of Los Angeles, but still officially in LA County, which matters for the story. And these were amazing programs by NSF, and they still exist to some degree, but not as big as back in the day. And basically it was, hey, we want to get kids exposed to science, and so have high school students apply to that program, and then they'll just match them up with a faculty member at some local university, even a community college, anything. And just once a week, they're supposed to go and just hang out around the lab. And then so they accepted me into that program. And I think then they realized that there was no college anywhere near me.
Oh.
And so my first science experience was them setting me up to work at the Catalina Island Marine Biology Lab off the coast of Los Angeles. So once a month as a 16-year-old, I would get in my parents' Cadillac and I would drive on a Friday afternoon in LA traffic. I would drive 3.5 hours across the entirety of LA County, then hop on a boat, go over to Catalina Island and sit in this marine biology lab in this marine sanctuary for a weekend doing stuff and then go back. And so I always say I got tricked into loving science because it was just this gorgeous location with kelp forests and Garibaldi fish and beautiful sharks and whatnot, and got to see what these marine biologists did. And there was a little bit of biochemistry that got me going, but it was enough to get me interested and say, oh yeah, this experimental biology is interesting. And pretty much pursued it continuously from there.
And then you moved all the way to the East Coast to do college, right?
Yeah.
And did you do research while you were in college too?
So at the time, there were really no research opportunities for high school students. And actually even for undergrads, there are only 2 places in the country that had undergraduate research programs as a normal thing. Undergrads could get into labs, and MIT was one of those too.
Mm-hmm.
And so I applied to MIT. I'd never seen the place. I'd never been to Massachusetts. But it was true. It was definitely a place where hands-on research was very encouraged, and you could get into labs and get exposed to world-class research even as a freshman, and start to get a sense of what types of that research you may be good at or like versus others. Yeah, so I did research throughout my time at MIT, and then went to grad school. Let's see, the research at MIT was on a couple of different areas, but essentially molecular biology.
So it wasn't immunology then?
No.
So did you apply to immunology programs? How'd you get into, or virology, immunology? How did you end up getting into that?
No, so I went to grad school at UCSF, and in the general, basically, biology program. Was it was the PIBS program. It was cell biology, biochemistry, molecular biology, genetics. My take was that molecular biology was basically great for everything and amazing. And so the important thing was to learn as much molecular biology as possible. And then there would be problems of interest, scientific areas of interest that one could apply those tools to. And at the time, immunology was very low on my list. Well, it wasn't on the list. I didn't go to UCSF for immunology or virology. Really, it was towards the end of my experience at UCSF. I helped out on a vaccine project in Raul Andino's lab. Yep. My main project was not my main. I was viral genetics, viral replication, and viral evolution. But I helped out on a vaccine project that was a monkey model vaccine project, and it turned out that the vaccine partially worked. And we had these as a group, there were these immune response measurements. So there were immune response measurements, and there was a partial success. And so then it was like, okay, we don't know any immunology. So we went and talked to all the immunologists that we could find around, and said, okay, what do you think? And they said, well, we don't know. That's vaccinology, not immunology. And that was to me, that was a profound experience, because I was like, what are you talking about? Like, I mean, immunology had clearly been this, and still is, in this extraordinary basic science field of research with all these amazing discoveries, and vaccines were this incredible and important area, and they just weren't connected, which didn't make any sense to me. And to some extent, that's for historical reasons, that vaccines actually predated immunology, and they were very empirical. But the fact that 20 years ago, 25 years ago, that was still the case. And so really it was that I said, all right, I'm going to work on that problem.
Connecting the two.
What's the basic underlying immunology, what are the principles underlying successful vaccines versus unsuccessful vaccines, that the process to date. So vaccines are the only medicine recommended for every single healthy person on the planet, right? Their economic return on investment is extraordinary. It's like one of the most valuable things one can invest in. And just the overall health and socioeconomic values of vaccines are really exceptional. But most attempts at vaccines fail, probably like 99% of them. And the ones that succeed, right, it's great, but surely we could do better at predicting which ones are likely to succeed or fail and be more efficient. And really, classically, it's you try a vaccine and it either works or it doesn't. And if there's anything intermediate, there's nothing to learn from. And so we've been trying to help out any way we could on that to really turn it into an engineering problem or a science problem. So that's really broad strokes the path of my career. So definitely experimental science early on, but really no inkling of immunology even when I started in grad school. For me, it was about, okay, what's a rigorous core knowledge base that I could learn that could be applicable to problems that come up that are of interest. Yeah.
So when you did find immunology, I have a favorite cell. My cell's the macrophage. You also have a favorite cell, and it's a TFH cell, right? I've heard you say this before. Or has it changed?
So TFH cells are my favorite cell. That's true. But I actually, broadly speaking, try pretty hard to avoid the favorite cell thing because
no favoritism.
There's, at least classically, there's way too much tribalism in immunology.
Well, that is true,
about T cells versus B cells, and you either like one or the other. And we definitely study T cells and B cells, and I generally don't get the tribalism because I'm like, the immune system has all these cells because they're valuable.
They're all important.
I trained in Rafi Ahmed's lab, which is classically a CD8 T cell lab, and I was basically the one B cell or CD4 person in the lab at the time. And there are definitely people in the lab who are like, B stands for boring. B cells are boring. Why would you bother? People only work on B cells if they're not good enough to study T cells. But yes, TF8 cells.
So
if I've got to have a favorite cell, TFH your favorite cell. Yeah, so it stands for T follicular helper cell. So it's or is that what you wanted?
Yeah. So can you tell us a little bit about what a TFH cell is and why it's important and why you care about it?
Yeah. So they're a subset of CD4s, and it connects with this overall arc of me being interested in, well, what are the fundamental immunological principles and ground rules, if you will, for successful versus unsuccessful vaccines. And the vast majority of currently licensed human vaccines work on the basis of protective antibody responses. Yep. So that does not mean antibodies are the only thing that can be protective, but certainly they've worked really well for these, vaccines that have saved millions of lives over 100 years. But we haven't understood well enough the ground rules for how you get a good antibody response versus not. And in fact, going back 50 years to the discovery of T cells and B cells as being different types of white blood cells, the first function defined for a T cell was T cells helping B cells make antibodies. Right. And yet for decades thereafter, it wasn't known what subset of those T cells provides that help to the B cell. So it was known that it was a CD4, but beyond that, it wasn't. And so that was one of the first problems that I worked on when I started my own lab. And we helped establish that T follicular helper cells are the cells that are essentially uniquely able to give help to B cells such that you get germinal centers. And so the vast majority of B cell memory, long-term antibody responses, and class-switched antibody responses depend on TFH cells. And that's why they're my favorite cell type.
And related to that in vaccines, let's go back a couple of years to 2020. So it's almost 5 years to the day when everything shut down here in New York. I don't know how it was where you were, but the schools closed. Yeah. Cornell closed. We were going to pivot to online teaching, and they said, no, we're going to pause everything temporarily. And it was kind of a wild time, but yet it was an incredible opportunity for immunology. So What were you thinking at the time? Did you realize the possibilities of what you've been able to do with samples that you collected at that time to study? You were one of the first labs, or the first lab, to show really a robust and deep dive into what was the immunology against SARS-CoV-2, and when the vaccines came out, what was the immunology of the vaccine? So what was going on then, and how did you decide to get into that, and what was it like?
Yeah, 2020, not the best year for humanity. But yeah, you're right. It was almost exactly a year ago today for the shutdowns. Right. So we with Alex Sete had the first paper to measure the coronavirus-specific T-cell and antibody responses. So the story there is, well, I think it was essentially exactly 5 years ago today. Sidney Ramirez, an infectious disease fellow at UCSD, so I'm at LJI, the La Jolla Institute for Immunology. We're a nonprofit research institute. We're on the UCSD campus, and we have a lot of interactions with the UCSD hospitals and clinicians. And so she had come to me about training in my lab. We talked in January. And then, 5 years ago today, she came and said, okay, I want to join your lab, do research in your lab, essentially as a postdoc while doing clinical work. And this coronavirus seems pretty interesting. I think it'd be good to work on that. I said, okay. I think you've got 48 hours before we shut down. And so you've got 48 hours to get an ID badge, go through biosafety training, go through human subjects training. And and we pulled that off. LJI is a very agile place. We just said, look, we need this done immediately. And so they did one-on-one, training and stuff. So then the shutdown happened, but LJI stayed open for COVID-19 research. So there were a couple of things. So there was a huge amount of fear and uncertainty about almost everything about COVID-19 at that time, including, did people make any immune response to the virus? Did people make a really strange immune response to the virus? And would a vaccine be possible, right, amongst other questions? And we felt like those are really big questions, and they'll probably take many years to fully answer. But that we felt like we could get a quick general answer by looking at the immune responses in people who had had COVID and not been hospitalized, right? They'd gotten sick, but they'd gotten better without needing a hospital. And then whatever immune response they had after they got better, well, something about that immune response cured them. And that should be informative about what it would take to get a vaccine, because that's what a protective immune response would look like. And Alex Sete, who's at LJI, he's the best person in the world at predicting and identifying human T-cell responses. And they had already made all these predictions for SARS-CoV-2, but they had no samples from people who had had COVID-19. And there were multiple reasons for that. But one was, for example, testing wasn't available. Right? The only way people were getting tested was if they were hospitalized. And hospitalized patients is a very different category. These are the people who had a lot of trouble possibly making an immune response. In fact, that's what we found out later, was that I think a lot of those are people who their immune response was slow and just not so good. We wanted to study people who got sick but got better without hospitalization. And so Sidney Ramirez then was able to go back and forth between us and the hospital and go find people who are coming to outpatient care, getting some of them to get good COVID tests that were trustworthy. That was another challenge at the time, right? Was getting a test that actually really told you, did they have it or not? And then bringing blood samples over and running those experiments. And we often get, in that paper, basically we showed that people consistently make T-cell and antibody responses. Those responses look pretty normal, and they don't look particularly big, and they look like the types of responses of essentially like you would make to flu or some new respiratory virus, and that they look consistent with what vaccine technologies were able to get. And so it was encouraging for vaccine development. And that paper was, we were really pleased that was so influential in the scientific community. It was influential in the medical community. All the major vaccine companies ended up citing that paper as this is the type of immune response that we're trying to recapitulate is what was reported in that first study. And people often ask us, well, how is it that this tiny little place in San Diego beat out, 1,000 other universities around the world at doing this first? It wasn't that we were lucky because we did it again for immune memory and did it again for a couple of other things. Part of it is having a lot of human immunology expertise in one building in an environment where we really had a long track record of already collaborating with each other. So it was just jump right in and get it done. We'd also collaborated a lot with people outside of LJI very successfully. And so when we called people and said, hey, we need this reagent or that reagent, they just sent it. It was not 6 months of MTA. It showed up 36 hours later because they're like, all right, do what you need to do. And we're sure it's going to all work out for us. But the other is actually LGI is actually really a place that's by scientists and for scientists. And so in the context of COVID-19, you talked about the shutdown, right? And it was shut down. Places, really closed. And some places would be like, well, we stayed open for COVID-19. And it'd be like, well, sort of, because there were a number of places that stayed open for the labs, but the core facilities weren't open. So they couldn't do the flow cytometry or they couldn't do the sequencing. They were hobbled, and we were able to say, look, we need this human subjects protocol figured out fast. It's just taking blood draws, right? It's not a big deal, but normally the paperwork would be several months, and instead we got it done in 4 days. And we said, look, we need the core facilities still open. And so those people were willing to say, yeah, we'll do that work. And then there was the biosafety question, right? This virus was clearly killing people. So you're like, well, you're going to have that virus in the building. What's the safe way to handle it? And we were able to go through that process quickly and safely to make it so we could make those findings and get them out, right, to the community really, really quickly. So we were really proud of that effort. So is the right team of people in terms of expertise? I think we did a really good job of asking the right questions or saying, look, we think the best way to do this is to look in people who haven't been hospitalized. And look, we don't know what immune response is best, right? I don't know if my favorite cell type is actually relevant or not, right? But we know the adaptive immune system has B cells, CD4s, and CD8. So we need to measure all three of those at the same time and just lay it out there, and see what looks like it makes sense. And then the third was really the more pragmatic structural things of being enabling that science to happen and happen quickly.
Yeah, you talk about happen quickly. One of the really impressive things about the science that came out, especially the immunology during that time, was it was so rapid. And you're talking about getting these protocols approved really quickly and things. Do you think that has changed at all how we think about how science can be done?
Oh yeah, for sure. It's basically March 15th, we started the project. We enrolled 20 people who had had the disease, and people who hadn't, and did all the immunological assays by April 15th, so 30 days later.
I see. Wow.
And I believe we did it faster than that. I have to go double check. And then Alex Seta and I analyzed the data and wrote the whole paper in 7 days.
Whoa.
Yeah, which is an incredibly different experience, right, from even what I'm doing right now. I mean, we just finished writing a paper that we just spent like 8 months doing the writing. And then submitted and published within a couple weeks. I think 14 days. Somebody was just reminding me that one was, I think it was online 14 days later. And of course, a key aspect of that, well, there are multiple key aspects, but one key aspect of that was we can say, look, here's the data.
Yeah.
Here are our thoughts on the data. The limitations of this are,
yeah.
We've done what we can to get it out there, but obviously we want everybody else to try and do similar experiments and see what they find, and double check and confirm these. And in fact, that paper is held up incredibly well, which we're very proud of. So yeah, science, there are definitely scenarios where science can be done quickly, and certainly one. I think one aspect that has held up is at least people thinking about what best practices are in the review process of papers.
Mm-hmm.
That it's definitely peer review is important. It makes sense to consider, does a paper need more experiments or not? But it's not that they need every possible experiment you could ever do and take an extra, year to publish the paper just for a minor thing that you could write in as a limitation instead, and instead get the data out there more publicly. And preprints definitely became much more standard and accepted in immunology and virology and molecular biology at that time, with all their strengths and weaknesses. And I think that was a big transition for people as well. For IRBs and things, yeah, I wish it had a lasting impact, but unfortunately,
not so much.
It hasn't. I think it showed that yes, you those things can move quickly.
Mm-hmm.
But honestly, most of the people who are on IRBs have, they've got all their full-time jobs anyways, and so it's just they're not paid, to full-time do that job, and so it's people finding a bit of time here or there to move something like that forward, and so now it's back to frequently. Those things can take quite a while to. So that's but yeah, there are certainly things that have been lasting, I think, positive changes in the research community from the speed of advancing science in that. And I mean, I am so proud of what the scientific community accomplished during COVID-19.
Yeah.
I mean, there's never been anything like it in the history of science, of how much knowledge was gained and how much accurate knowledge was gained about a new virus and a new disease and new therapeutics and new vaccines, right, within a single calendar year, was just amazing. And how much people work together to accomplish that is, really mind-bendingly great.
Yeah, when you have that common threat, a goal, people will band together and work together really carefully and closely. I wish that we could do more of that going forward, but I think that it's going to end up being the little fiefdoms again, and competition is going to come back into things a little bit more like it was before. But maybe we learn to be a bit more collaborative than we were before. I don't know. It'll be interesting to see how it all pans out. I found it really interesting that some people were posting on social media data, right? So they could get it out that fast so that other people could take advantage of it. I mean, that is just unprecedented in science as well. Yeah. It was a very interesting time.
Yeah, that was exceptional. And there was real value there. I definitely gained value from those things. Collaboration and competition. I'm a big believer in both. I love collaborating. Many of our most exciting papers and most influential papers have been collaborations with people who are amazing scientists and have expertise that I don't have, and can generate great new things as a result of collaborating. At the same time, science is predicated on competition.
Yep.
And so I think, while it's not lovely, it's necessary, and it's important to keep in mind. Actually, that was I learned that when I wrote this biography of David Baltimore and was interviewing him, and people around. This is when I was 20 years old, and a number of these Nobel Prize winners and others were like, "Look, competition's just it's necessary. The way you get scientific advancement is who can make the discovery first and who can publish it first," right? Going all the way back to the famous example of Darwin, that Darwin had discovered evolution and he had this all figured out, but he didn't publish it for 20 years or something. And he only finally published it when there was competition to actually make it public. That tension is necessary. And at the same time, collaboration is so powerful.
Yeah, it's a tension there between the two. Do you have, so that's a really good piece of advice for people going into science and for trainees. Do you have other suggestions or advice that you give to trainees who are starting on their path in science?
Yeah, my number one advice to trainees is find experiments you can do quickly. Because I'm a big believer in failure as a great teacher.
Yep.
And I had the good fortune of doing my PhD with Raul Andino on poliovirus, mm-hmm. which has an 8-hour life cycle or 24 hours to do an experiment. So as a result, I could do an experiment every single day. And I could screw up 29 experiments in a month and still have a successful experiment, And I mean, those failures were really powerful teachers. They'd be like, oh, I thought this was the right control, but actually, I needed to do it this other way, or, all kinds of different ways of failing and learning how to be a good experimentalist. And there are lots of experiments you can't do like that, right? They're just big, expensive, long experiments, but and doing those is great and valuable, but from a learning perspective, it's good to have at least some project or small thing, that you can do more rapidly and learn how to succeed through failure. Yeah, that's my I'm a big believer in that.
That's good advice. So I have a couple of last questions I want to ask you that are kind of fun. So what's something surprising that people, if that they don't know about you?
Oh, you should probably ask my wife. I don't know. I'm a military kid. I grew up in the military. Okay. My dad was in the military. I lived 11 different places before I went to college. Yeah, that's one.
Is that why you were in the desert outside of California?
Yeah.
That's where you were stationed?
Yeah. Uh-huh.
Yeah. Oh. How do you think that being in the military has shaped how you approach science?
I don't know, right? That's one of these things you talk about with childhood, you know? You only get one childhood, so you don't...
Yeah.
I don't know what it would have been like to live the same place for 18 years as a kid. It just didn't happen.
Do you think that adaptability and that flexibility and being able to,
people
try
talk
new
about
things?
That. It could be. Certainly when I applied to college, I really didn't think a whole lot about the fact that I hadn't visited the place and didn't know anything about it besides literally a photo and a brochure. Plus, what I said, the write-ups of, oh, well, that's a place where you can actually do research as a college student.
Yeah.
I'm like, all right, that's all I need to know. The rest of it will kind of work itself out.
Yeah.
Assuming I could actually get in. I think it's plausible, but.
Interesting. So I also like to ask, why do you do what you do?
And
so
I will say, growing up in environments like that, you meet a lot of different people in a lot of different contexts. I grew up in the Florida Panhandle and in Alabama and then in California and in England. A lot of different, and you just meet a lot of different folks with a lot of different experiences and perspectives, and that, that certainly was of value. Sorry, you were going to ask.
Oh, no, that's okay. No, I like to ask, why do you do what you do? What is your passion? What is your reason for doing science and immunology?
I think that's a fantastic question because science is really hard. So, I think I don't think you can't really do it unless you're passionate about it. You gotta be doing what you love because we work on hard problems where you can easily go a whole month and every day is experimental failures. And we've definitely had projects where just for 6 months, right? There's just no light at the end of the tunnel for those things. So you really gotta believe in what you're doing and be passionate about it to be able to get through those. And you're working at the edge of knowledge, right? These are hard problems. I certainly just have a basic love of science and new data, but I am really passionate about vaccines. I founded my lab to work to better understand vaccines because they're some of the most valuable medicines in the world. They've saved more children's lives globally than anything besides clean water. Just the impact of existing vaccines and new vaccines is extraordinary. So if I can help even in small ways to make those more likely to be successful or faster in the future, that I'm passionate about that.
That's great. So my last question to you is, if you weren't a scientist, what would you have been?
Ooh, that's a good question. These are good questions. I don't know. Oh, well, I'd be a writer, right? Because I ended up double majoring in biology and in writing, and I've published a book. It did pretty well. So yeah, I would probably do. So science writing is probably what I would. Well, I think. whatever.
I was going to say that's a bit of a cheat because you are actually a writer too, because all the papers we write are writing.
Yes, that's true.
But you combine the science and the writing, and it's perfect.
If it wasn't one of those two, yeah. Yeah, I don't know.
That's great. So thank you. Really appreciated this. So
thanks.
That's a special episode of Immune, an immune booster, the scientist behind the science. If you have questions and comments, please send them to immune@microbe.tv. And if you enjoy this, please support the shows of Microbe TV at microbe.tv/contribute. Today was joined by my special guest, Shane Crotty from the La Jolla Institute for Immunology. Thank you so much for joining me.
Thanks for having me. This is great.
Great. I'm Cindy Leifer, and I'm Cindy Leifer on Blue Sky. The music for Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.